How is it that your brain can understand all of the sounds entering your ears right now? And how can you understand all the things that you currently see? How do sound waves or light waves get turned into something that the brain can understand? In this section of the biological basis of psychology, we're going to take a look at the transduction of vision, and then we'll move on to all of the other senses, like hearing, taste, smell, and even touch. But let's start today with vision, because visual transduction is a very important process, and it's probably the more complicated of our five senses, because we have many different parts of the eye that are responsible for the visual process. As information enters into your eye via the front of your eye, where your pupil, iris, cornea, and lens are, it's going to pass that information, those light waves, to the back of your eye, where your retina, your fovea, and your optic nerve are. The retina on the back of our eye is where the light waves fall. It contains a wide variety of cells known as photoreceptors, which are also called rods and cones, that begin the process of transducing all of the visual information. There's only one spot on the back of our eye where there are no photoreceptors, and that is referred to as our blind spot. It's where the optic nerve converges together to exit the eye and take the information up into our brain. And so because of that, it's impossible for us to have any visual cells there. You don't notice your blind spot though, because our brain does a great job of covering it up for us. On the front of the eye, one of the key players when it comes to vision is the lens. The lens is what actually changes shape in order to allow us to focus the information that is landing on our retina. That changing of shape is referred to as accommodation. Now, when we need to focus on something, our lens will accommodate to allow us to see clearly. But because of changes in shape of our eye and our lens, some people will not be able to experience things in full focus. People that experience myopia or nearsightedness happens because the accommodation can't take place if the eyeball is too long. Or farsightedness, also known as hyperopia, takes place when the eyeball is too short. It prevents the correct accommodation to take place to allow us to focus information into the back of our eye to see clearly. Glasses and contacts help greatly with this to allow us to be able to see. The photoreceptors, being our rods and cones, are going to take that information to be able to start the transduction process. Rods are responsible for black and white. They exist mainly in the peripheral part of our vision, and they're what are activated at night. If you notice at night when it's dark out, you don't see things in as clear of color as you do during the daytime. Cones are the other type of photoreceptors in the back of our eye, and they are responsible for color. There are blue, green, and red cones in our eyes that are going to activate when different light waves hit them to allow us to see a wide array of colors. Speaking of color, there are two color vision theories that help us to understand how we see the wide variety of colors that do exist in the world. The first one is called the trichromatic theory, or the Young-Helmholtz trichromatic theory, and this goes along with those three colored cones in our eyes, that different light waves are going to produce different combinations of colors to allow us to see the wide variety of colors that we can see. The opponent process theory takes that a step further and looks at color from the ganglion level, which is further into our eye as that information is going to be passed into our brain. The opponent process theory says we have three types of ganglion cells that are responsible for color pairs: red and green, blue and yellow, and black and white. And when you stare at a color for a very long time, those cells continue to fire over and over again. Eventually they are going to become tired. And as those cells start to wear out, if you look away very quickly, you will see a negative after image of the other color in that color pair. So for example, we see here this image of a Canadian flag that is green and black. If you stare at that Canadian flag as you hear me talking, your eyes are going to continue to fire off those colors of green and black, and they'll start to fatigue. So in a moment, what we'll do is remove that flag, and you might see for the briefest moment an after image of a Canadian flag in red and white. That negative after image is proof or evidence that color is also processed at the ganglion level. These ganglion cells then converge together to form the optic nerve. They'll exit out the back of our eye and take that information up into our brain so that our brain can process the visual information. And that is really part of the entire visual process as well. Now, sometimes there's something that goes wrong in the color vision process. Dichromatism, for example, is a type of color blindness where you only have two types of functioning cones. Typically what we'll see is either red or green cones that are not functioning normally, and so those are referred to as red or green color blindness. If you look at the image on the screen, inability to see that would be a result of one of those cones malfunctioning. Monochromatism is where only one type of cone is functioning, and so you're really not able to perceive any color. In monochromatism, you can really only see in light and dark, or black and white. And there are also some really unique features that take place when vision makes its way up to our brain. Prosopagnosia is a great example of what happens when visual information is passed up into our brain and whether or not it functions the way it's supposed to. As your brain takes this information to our thalamus and our visual cortex in our occipital lobe, it will also pass it to other areas of the brain, such as the fusiform gyrus, which is helpful in identifying faces. So the fact that you can see my face and know who I am comes from your functioning fusiform gyrus. If it's not functioning properly, even though you can visually perceive the face that's in front of you, you wouldn't be able to recognize it. It's the part that kind of brings all of the features together for you to be able to recognize faces. Last but not least, when we talk about vision, we are referring to light waves and how those are entering into our eyes to be passed on to these photoreceptors. Wavelength is what determines the color that we see via our cones. Different distance between those wavelengths, whether they are close together or further apart, are going to create the different colors that we're able to see. The amplitude of those waves can also affect the brightness of the colors that we see. It's also what impacts the loudness of sound when we're talking about hearing. So that's what we'll talk about next, is how sound waves get transduced in our ears to help us to make sense of the sounds that we experience in the world around us. We'll stop there for now, so thank you so much for watching, and as always remember, be kind to your mind.